The overall goal of this experiment is to detect retinoblastoma protein SUMOylation under endogenous and exogenous conditions in human cells. Small ubiquitin-related modifier, or SUMO, family proteins are conjugated to the lysine residues of target proteins to regulate various cellular processes.
Usually, it's more challenging to study protein SUMOylation in cells due to the complexity of SUMOylation in vivo. This experiment detects the SUMOylation of both endogenous and exogenous RB proteins. Though this method can provide insight into the SUMOylation of RB protein, it's also appropriate for the detection of many other SUMO target proteins in cells.
To begin, add 25 milliliters of DMEM containing 1% penicillin streptomycin and incubate the HEC293 cells at 37 degrees Celsius for 72 hours. Wash the cells off the plate using 3 milliliters of ice-cold PBS. Then transfer the cells into a new 5 milliliter tube. To obtain G1 phase cells at the end of the G0 synchronization process, remove the DMEM and add back fresh growth medium, thus allowing the HEC293 cells to re-enter the cell cycle. Then collect the cells at different G1 phases for cell cycle analysis and further SUMO assay. To synchronize the HEC293 cells at the S phase, use the double thymidine block method. First, grow the HEC293 cells to a confluency of 50%. Then wash the